This lecture will cover the first part of Chapter 9. Chapter 9 is all about the autonomic nervous system, so let's quickly review how we got to the autonomic nervous system. So if we take the nervous system as a whole, we can divide this into the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is composed of nerves and ganglia. So recall that nerves are just a bundle of axons outside of the central nervous system, and a ganglia is just a cluster of cell bodies in the peripheral nervous system or outside of the central nervous system. And our peripheral nervous system, we can further divide, and we have our somatic division. So we can have, somatic sensation. This is coming from the skin or special senses, regarding temperature, touch, and pain. I feel this. And then we have somatic motor. And somatic motor is going to skeletal muscle, or voluntary muscle. Then we have our autonomic division. So this is going to visceral structures, the motor portion, and this is involuntary. So what we're not consciously thinking of, breathing, heart rate, things of this nature. Okay, so we're going to get sensory information regarding our body's homeostasis. And then we have visceral motor or autonomic that is going out to maintain that homeostasis. So if we say, visceral motor, let's review what our visceral structures are. Visceral structures are cardiac muscle, smooth muscle, or glands. Something else to review from way back at the beginning of Chapter 7 is that visceral motor is going to involve a 2-neuron sequence. Okay, so the neurons, they communicate with one another at this area of the synapse. And we can see, that there's a cell body here. So we're going to have a cluster of cell bodies, which we know is a ganglia. So here we have an autonomic ganglia. Now we can name our neurons relative to the synapse or the ganglia. So before the synapse or the ganglia, we have the preganglionic or presynaptic neuron. And the preganglionic or presynaptic neuron, the cell body is going to reside within the central nervous system, and then the axon is going to extend to the ganglia. Then you're going to have a synapse with our next neuron. So this is after the synapse. So we call it a postganglionic neuron. And then the axon of the postganglionic neuron is going to extend to our effector, which again is a visceral structure, so cardiac muscle, smooth muscle, or gland. Now let's do a side-by-side comparison of the somatic division and the autonomic division. So over here we have our somatic division. So we have a sensory receptor in the skin, and it's taking up sensory information regarding temperature, pain, touch, proprioception. You see it coming in. Here's our cell body, axon, and it's going to the central nervous system. It's taking that information to the central nervous system. Now we have somatic motor, which is going to skeletal muscle, and we have our cell body in the anterior horn of the spinal cord, and the axons extending out through the ventral roots over to the spinal nerve and to the effector. Now, if we come over and look at the autonomic nervous system, right, the sensory information is coming in regarding our body's homeostasis, okay, and the sensory pathway is still consisting only of one neuron, again, our cell body, taking the dorsal roots back to the dorsal horn. So now where it gets really different is with visceral motor, the outgoing command. Okay, so now we can see our two neuron series. So our preganglionic neuron, the cell body, is in the lateral horn. And we can follow our axon coming out the ventral root. And now this pathway is going to be a little bit different depending on if we're talking about the parasympathetic division or the sympathetic division. But now let's just focus on the two neurons. So our preganglionic neuron is leaving the central nervous system, and it's going to synapse with our postganglionic or postsynaptic neuron, which we can then follow to the target or the effector, which is a visceral structure, so cardiac muscle, smooth muscle, or a gland. Now let's compare the two divisions of the autonomic nervous system. We have the sympathetic division and the parasympathetic division. So in short, and we'll go through different effects of each division, but in short, the sympathetic division is preparing the body for physical activity. Okay, so this is our fight or flight response. What are we going to do if there is a bear, right, charging into the room at us, right? We're going to be prepared to, right, fight for our lives or get out of there as fast as we can. So we can think of the E's, right? Emergency, exercise, excitement. Okay, so things that are amping us up, right? Increase heart rate, increase blood pressure, dilation of the pupils, things preparing the body for physical activity. This is also called our thoracolumbar division, and this is pertaining to the preganglionic neuron. Where is the cell body of the preganglionic neuron? Well, we can just look. Thoraco, okay, so thoracic, lumbar, okay, lumbar, thoracolumbar region of the spinal cord is where we're going to find the cell body of the preganglionic sympathetic neuron. Now let's go look at parasympathetic. Okay, so parasympathetic is called our rest and digest division. Okay, so this is going to stimulate the involuntary activities while we're at rest. Okay, so if we think about it, right, if you're in sympathetic mode, if you're in emergency mode, right, this bear is chasing you. Does it make sense for the body to waste energy digesting the food that you ate? No, of course not. So the digestive system under sympathetic stimulation gets turned down. But we have escaped the bear, and now we've gone home and, right, we're relaxing, gonna watch something on Netflix, whatever, and have a big meal, right? So we're nice, and now we have time to activate our digestive system. So the parasympathetic division is going to increase digestive activity and sort of slow everything else down, right, in time for rest. Now this is also called the craniosacral division, and just like with the sympathetic division, this is pertaining to where we're going to find the preganglionic neuron, or the cell body of the preganglionic neuron. So cranio, we just learned, right, that cranial nerves have a parasympathetic function. Okay, so cranio means cranial nerves. And if we review, we have cranial nerves 3, cranial nerve 7, cranial nerve 9, and cranial nerve 10. Oculomotor, facial, glossopharyngeal, and vagus, which all have a parasympathetic function. And then sacral is referring to the sacral portion of the spinal cord. I'm going to take some time and go through the different responses that we would expect to see from different effectors within the body under sympathetic stimulation and under parasympathetic stimulation. But while we go through this, right, think about it logically and make it make sense. Don't just memorize different effects because it will be overwhelming, right? Think about what's going to happen to your body if, right, there is a bear or an elephant or something chasing you and you're running away trying to get away from it versus what's going to happen to your body if you're just relaxing on the couch reading a book. Okay, so we can make a chart to sort of organize everything, and we can say what's the effector, and then we want to compare parasympathetic stimulation versus sympathetic stimulation. So first, let's think about our heart. When we're running, right, we're going to need an increased heart rate. We need more blood getting out to our skeletal muscle, right, because it's going to be, right, carrying us along the way. So in sympathetic, we're going to have an increase in heart rate versus parasympathetic. If we're just lying down looking through a magazine, right, we have a much lower heart rate. So we have a decrease in heart rate. Now going to our smooth muscle, our visceral structures, smooth muscle, we know lines are blood vessels. So if we have a blood vessel, right, blood's traveling through, it's lined with smooth muscle. If that muscle contracts, we're going to have vasoconstriction, which is going to decrease the diameter of the blood vessel, and we're going to have less blood going to the area that that blood vessel is traveling towards. And then if we have that smooth muscle relax, we have vasodilation, so the diameter is much larger, so we have more blood going to that specific area. Okay, so if we have blood vessels going to skeletal muscle, well, if we're running away from something, right, something's chasing us, we're running, we need more blood getting to that skeletal muscle, right? We need more oxygen getting there. So we're going to have vasodilation. Okay, but so we're not really going to have a parasympathetic response here, and we'll talk about that in a bit. But let's look at some other blood vessels. What about blood vessels going to the skin? Well, if we're in duress, it's not really important that we have a lot of blood flow going to the skin. We we need to send that blood elsewhere, right, to our skeletal muscle. We are running. So now, right, we can sort of constrict that and reduce blood flow going to the skin. It's not important when we're under sympathetic stimulation. Okay, so now we're going to have vasoconstriction. Okay, and we can say the same thing. What about the blood vessels going to the gastrointestinal tract? Well, we already said it's not important to be digesting food if we're right, if we're running away from something, if we're in fight or flight mode. So blood vessels going to and same with the kidneys, right? The kidneys filter blood and make urine, but that's not really important if we're in under sympathetic stimulation, right? We're out of here or we're fighting off whatever is threatening us. Okay, so we don't need a lot of blood going to the GI tract. We're not digesting. We're not resting and digesting like we would be with parasympathetic. Okay, so again, here we're going to have vasoconstriction, reduce the diameter of that blood vessel, less blood flow to these structures that are not really active when we're in a sympathetic response. Okay, so with that, with our gastrointestinal tract, this is also lined with smooth muscle that's going to control the movement of substance that we eat, of food stuff, through the gastrointestinal tract. So when we're under parasympathetic stimulation, resting and digesting, right, the parasympathetic division is going to stimulate motility within the digestive system. Versus in sympathetic, we're going to have an inhibition of that movement, right? It's not important. And what about our salivary glands, right? Saliva, that's going to help us start breaking down the macromolecules that we eat. Okay, so these are going to be stimulated by our parasympathetic division, which we discussed when we were talking about cranial nerves, because we have, right, cranial nerve 7, the facial nerve, that stimulates the sublingual gland, submandibular, as well as the lacrimal gland. And then we have cranial nerve 9, glossopharyngeal, that stimulates the parotid gland, which is a salivary gland. Moving to our airways. Our bronchioles, our airways, are also lined with smooth muscles. So we can have the same thing. We can have bronchoconstriction, so we're reducing the amount of air that we're bringing in, or bronchodilation, where we're increasing that diameter, allowing more air in. So what about okay, so if we're lying down, relaxing, reading a book, do we need this massive amount of oxygen coming in? Are we breathing heavy? Are we, right, fleeing? No. So we can have bronchoconstriction. Versus when we're in fight or flight, right, we're going to open those up. We're taking larger breaths. We need more oxygen coming into the body. So we're going to have bronchodilation. Okay, then our kidneys themselves. Okay, so we're going to have a sympathetic response on the kidneys, and the kidneys are going to be stimulated to release renin. So we'll talk about renin a lot through this semester. So for now, we can just say, right, stimulates, the release of renin. And long story short, renin is going to increase our blood pressure. All right, so the liver. Now, what is in the liver that we could possibly need if we need more energy, right? We know that glucose is stored in the liver as glycogen. And we can break into that glycogen and liberate that glucose into the blood when we need additional glucose for energy. So when we're under sympathetic stimulation, the liver is going to stimulate glycogenolysis, right? Breaking down glycogen and releasing glucose. But if we're in rest and digest, then we don't need that extra glucose. So we want to store it in the liver, right? So parasympathetic stimulation is going to stimulate glycogenesis, right? Taking glucose and storing it as glycogen. Okay, then we can say our bladder. Okay, rest and digest is going to include, right, the kidneys creating urine and then our body getting rid of that urine. So when we release urine, right, to outside of the body, this muscle of the bladder is going to contract. Okay, so we're going to get contraction of the bladder so that we can urinate. Under sympathetic stimulation, well, we don't want to be doing that. We don't have time for that. It's not important right now. Okay, so that muscle is going to relax. So there's more, right, space in the bladder for urine to occupy because we're not concerned with getting it out of the body. Okay, then we have erector pili muscle. So these are little pieces of smooth muscle that are attached to the hair on our body. Okay, and these, again, we're not going to have a parasympathetic response, but when we're under sympathetic stimulation, we're going to have contraction of these little muscles that's going to pull our hair up. So think about a little kitten or a cat, right? When they're fearful, they fluff themselves up and make themselves look bigger. Well, we do the same thing. We just don't have a fur coat that gets big. So, but same principle. This muscle is going to contract and pull our hair up to, right, make us look bigger. It also is what will give us goosebumps. Okay, so we have contraction of the erector pili muscles. and our sweat glands. Okay, so when you're nervous, when you're right in under sympathetic duress, when you're fight or flight, notice that right there's secretions from your sweat glands. Okay, so under sympathetic stimulation, we're going to have secretions from our sweat glands. A few more effectors. We can talk about the pupil. And similar to what we saw with the airways or our blood vessels, right? We can either constrict the pupil, which allows less light into the eye, or dilate the pupil, which is going to allow more light into the eye. So in fight or flight response or in sympathetic response, right, we are going to see dilation of the pupil and right, more light coming in. We can see what's coming at us, what is, causing us to be in this fight or flight response. Versus in parasympathetic stimulation, we have constriction of the pupil, allowing less light into the eye. And to review, this is done by cranial nerve 3. I already kind of mentioned the lacrimal gland when we were talking about cranial nerve 7. But again, the lacrimal gland is a gland up here in the eye that releases tears that will sort of lubricate the eye. Every time you blink, they sort of roll the tears across the eye. So this is going to be stimulated by cranial nerve 7, the facial nerve. Okay, so which we know is involved with the parasympathetic division. Okay, then we can move on to the adrenal medulla. So without going into too much detail, the adrenal gland is right on top of the kidney. Okay, this is not proportional, but that's okay. And the adrenal gland is divided into two regions. We have the cortex, the cortical region, which is this outer portion, and then the medulla, or the medullary region, which is the central portion of the adrenal gland. Okay, and this central portion of the adrenal gland, the adrenal medulla, contains norepinephrine and epinephrine. So norepinephrine, epinephrine can be neurotransmitters within the sympathetic division of the autonomic nervous system. They can also be released as hormones into the blood to have a more prolonged systemic effect. So under sympathetic stimulation, we're going to have stimulation of the adrenal medulla to release norepinephrine and epinephrine. And then lastly, we have our adipose tissue. And we know that adipose tissue is going to store triglycerides, and these triglycerides can be broke down into glycerol and fatty acids and used as an immediate fuel source. So do we need this fuel source if we're resting and reading a book? We do not. But if we're under sympathetic stimulation, then we want, right, we need more energy, we need more fuel. We're going to be fighting, or getting out of there. Okay, so from our adipose tissue, under sympathetic stimulation, we're going to have the stimulation of lipolysis. So those were differences in effector responses between parasympathetic and sympathetic. We're also going to talk about the different pathways that these neurons can take and different neurotransmitters and different receptors to lead to these different responses that we saw between parasympathetic and sympathetic division. But to best understand the pathway, let's talk about some of the anatomical differences between the sympathetic and parasympathetic division. So overall, we have three major differences between the two divisions. One being where the preganglionic neuron, or the cell body of that preganglionic neuron, is. Okay, so we know it's within the central nervous system. In the parasympathetic division, can either be in the brainstem as a cranial nerve or in the lateral gray matter of the sacral region of the spinal cord. Okay, so this is where we get the name craniosacral. In the sympathetic division, it's in the lateral gray matter, T1 to L2, which is where we get the term thoracolumbar. Now, our second major difference between sympathetic and parasympathetic, or our second major anatomical difference, is the length of the preganglionic and postganglionic axons. So in the parasympathetic division, our preganglionic axon, we know the cell body is in the brain or sacral region of the spinal cord, and then the axon is going to be very long compared to the postganglionic axon. So the postganglionic axon is going to be quite short or shorter versus the sympathetic division where we see the opposite, and we have relatively short preganglionic axons, but then longer postganglionic axons. And then our third major anatomical difference is the location of the ganglia. Okay, so where is the synapse going to be? So in the parasympathetic division, the autonomic ganglia are usually very close or in the effector organ, versus in the sympathetic division, the ganglia usually, we're going to look at some different pathways, but for the most part, the ganglia are located right next to the vertebral column in the structure called the sympathetic trunk or sympathetic chain. We will start with the sympathetic division. We've already pretty much covered what happens with the parasympathetic division when we talked about cranial nerves. We know cranial nerves 3, 7, 9, and 10 are involved in the parasympathetic division. So first, let's go through the different pathways that we would expect in the sympathetic division based on what effector the neurons are going to. Okay, so the sympathetic division, right, or the thoracolumbar division, we know that those preganglionic cell bodies are going to be in the lateral horn of the spinal cord, T1 to L2. Then the ganglia, right, the autonomic ganglia, where sometimes we'll have a synapse, these are going to be close to the central nervous system, right, because they're next to the vertebral column, so they're close to the spinal cord in the sympathetic trunk or sympathetic chain. Okay, however, with the sympathetic division, we have 4 different pathways depending on what type of effector the neurons are going to. Okay, so we'll look at these different pathways. We have 4 sympathetic pathways. So here in this picture, we can see our spinal cord in the back within the vertebral column. And then we can also see this structure called the sympathetic trunk and swellings within the sympathetic trunk that are called sympathetic ganglia. So if we follow this sympathetic trunk down, we notice that there are these big swollen regions. Now let's again define the term ganglion, right? This is this enlarged region because it's a cluster of cell bodies in the peripheral nervous system. Oops, that's an N. Okay, so we're talking about the sympathetic division. Our preganglionic sympathetic neuron is going to start thoracolumbar, so in the lateral horn of T1 to L2. Okay, so there's our preganglionic sympathetic cell body. Now the axon is going to leave through the ventral horn, and it has to get to the sympathetic trunk. So how is it going to do that? Well, we can see we have these little connections over to the sympathetic trunk. So these are called the rami communicans, and we have a white ramus communicans and a gray white ramus communicans. And the difference is whether the axons are myelinated or not. So we're going to head over, and now we're to the sympathetic trunk. Okay, so we have these sympathetic ganglia. We know there are cell bodies here. So in some pathways, the synapse is going to happen in the ganglia. In other pathways, it won't. But let's use that as an example. So this preganglionic sympathetic neuron has taken a ramus communicans, got on the sympathetic trunk, and now it's going to go down and synapse with the postganglionic sympathetic neuron, which can do a number of things depending on what the effector is. But let's just say, for this example, okay, he's going to take the ramus communicans and then go off towards the effector.